Next speaker, Desmond O's from UPenn, who's doing a lot of work, TMS in combination with fMRI for neuropsychiatric disorders, and so really looking forward for his presentation.
Thanks for having me. I'm sorry we can't all hang out together and talk about brain stimulation. Hopefully at a future meeting we can all get together. But I'm glad to be here in any forum, in any format, to talk to you guys. So I'm going to talk to you a lot about TMS fMRI. That's primarily what my lab does, and I think that it can show us some really important things. Some of them we've shown already, and some of them are works in progress. So I look forward to telling you more about this work. All right. So one of the motivations for combining fMRI with TMS is that if you look clinically where we stimulate for treating depression, you're kind of hoping based on a single measurement that you land in an area of the brain that based on the head size, you're actually not hitting in a lot of cases. And so I think this group especially is receptive to the idea that imaging might inform TMS. And I think moving that into the clinic is an important goal. So another more relevant to the imaging kind of folks point is that if you take a group average from an atlas and you say, hey, I think I know where the hotspots are for this network or that network, it doesn't always map on very well to the individual subject. So if you look on the left, this is an average of a working memory task, a very common one. And then on the right, you see where are strong activations proportionally by the percentage of individual participants showing a strong activation there. So you can see that the maps are smaller, and sometimes they go away entirely. And so the group average is not always very representative at the individual level, is the point. That's been shown also by Mike Fox's group, suggesting the subgenual connectivity is a good treatment target for depression. And if you use a group atlas, it works for some subjects, right? So the peak from the group map on the left shows the same sort of relative weight in a single subject in the middle, but then individual subjects can actually have very different or opposite signed connectivities, which presumably would give you a suboptimal clinical response if you just use the group average. So luckily the technology is there to do neuronavigated TMS to know what area of the brain you're stimulating, and you can take a variety of approaches. You can take resting connectivity, like in Joel's talk. You can try a group atlas, you can look for a structural kind of zone, diffusion, imaging, whatever you think is a reasonable map for a brain network, and have that online available to you from taking scan data, right? Another important kind of mention for doing Interleaved TMS fMRI is that you know, if you're an fMRI experimenter, that the fMRI bold signal takes a while to really maximize its signal, and having that sluggishness and timing is a bit frustrating in some cases, but in the case of interleaving TMS pulses with an fMRI recording, it's really ideal because you can place gaps between your readouts of the signal where you can deliver the TMS, and then you can measure the brain-wide response to that TMS pulse without corrupting the signal. So it's really convenient for us. And what you end up with is designing an fMRI run with interleaved pulses of TMS, where you do that a bunch of times and you average those out just like you would any other fMRI event, and you can make a map of the brain. This time, it's instead of looking at correlations to a task or something like that, you can actually measure the causal influence of a TMS pulse at a particular location and measure its brain-wide response. So that doesn't mean it's necessarily a direct pathway, but it's in the downstream causal pathway. And so I think that can tell us a lot about which nodes at the surface that we can stimulate with TMS are communicating with deeper or more remote brain areas. So an initial foray into this work, this is from work that I did at my old lab at Stanford, where we just take some canonical resting state networks and ping them with single pulses and say, can we get the network to respond to TMS, not just the node that we stimulate? And we were able to show that that's happening in salience network, central executive network. And we also did a little neuromodulation to watch how connectivity changes in response to 1 Hz stimulation. So that was the first initial kind of try at doing Interleaved TMS fMRI. And since then, since starting my lab at Penn, I've been trying to find pathways that can really sort of hit things that are relevant to anxiety and depression, and through collaboration, working memory, and other cognitive processes. But the focus of some of my initial projects is saying, hey, I think there's a subgenual cingulate kind of abnormality in depression. There's an amygdala abnormality that keeps showing up in fMRI studies of anxiety. Let's see if we can actually influence those and show that we can influence those by pinging those circuits and mapping them with Interleaved TMS fMRI. So the first, this is basically a pilot study that I'm going to be telling you guys about. So it's taking a bunch of subjects, but stimulating lots of different hotspots based on resting connectivity, seeding the subgenual cingulate and the amygdala, and seeing if we can get evidence that we can move those targets. So the subgenual cingulate and the amygdala are deep in the brain, so any kind of evidence that we can influence them with TMS would be a really important stride in kind of possibly developing more kind of targeted treatments focusing on specific brain regions. And so we were able to show that. This is a sample of 14 healthy subjects, and we were able to show that hitting the hotspot for the amygdala on the surface of the brain, we were able to move a couple of the subregions of the amygdala. So this is the superficial and central medial amygdala, the 2 right-hand box plots. So those moved significantly, activated those. And then we also, that crosshair is at the peak activation for the whole region. And that has, according to the Ulick Atlas, a decent kind of probability of being in all 3 regions. So it was fairly non-discriminating in activating the amygdala. So similarly with the subgenual cingulate, we were able to move the region of interest, so generating a negative bold response in the subgenual, which would be presumably the clinically useful direction. We also found evidence that a subgenual network that we defined ourselves using the same pre-processing pipeline in a larger healthy cohort also moves off its target. And you can see the error bars are a little tighter for the network. So we were able to kind of move that even more cleanly than we could the region of interest. And to show that there's, even if you kind of take out some of the regions that are overlapping with the DMN also shows a negative bold response, but not to the same degree as the network or the specific region, the subgenual. Another thing that we wanted to play around with a little bit is I showed you that, that map of all the stimulation points. And so we ended up hitting a lot of different kind of cortical zones in standard space. And so we wanted to go back and say, well, all right, we hit a bunch of these zones. Does it actually matter, not just hitting a resting connectivity peak, but also what zone of prefrontal cortex you happen to be stimulating in? And so in the case of the amygdala, it was true that actually it matters quite a bit what zone of prefrontal cortex you hit with TMS and the direction of the amygdala response. So if you're in motor cortex or inferior frontal zones, you get a negative bold response. And if you're in the anterior middle frontal or superior frontal zones, that's where the positive activation was coming from. So with the amygdala targeting, this informs us to not only take the peak based on resting correlation, but also pay attention to what zone of the cortex you're in if you want to move the amygdala in a specific direction. When it came to the subgenual cingulate, it actually didn't matter at all which zone you stimulate in. As long as you're hitting one of the subgenual resting connectivity peaks, you get that negative evoked bold response. So it's really nice to show these pathways and show the kind of mapping that we can reach these distributed brain regions, but we're also playing around with the functional relevance. We say, okay, well, let's hit these targets in a clinical context or more clinical context, and tasks that we think are relevant to the disorders that are kind of abnormal with these pathways. So the first thing we did is just look at baseline ping of the circuit. Is there any relationship with trait-like symptoms that people are reporting? And so at baseline, the more negative your evoked response, the lower your negative affect score. So this is an depressed sample, suggesting that the strength of that pathway is related to their reports of mood symptoms. And then stepping up to an actual neuromodulation, I think, is the next level of complexity, but also something that we're showing some promise in. So let me break this down for you guys. This is, we take the depressed individuals and we give them a kind of mini treatment just to test neuromodulation of the circuit and to see if there's any change in their symptoms, any change in their brain responses to TMS. So far we've just looked at the symptoms so far, and it looks like just doing 3 days of theta burst to a depressed patient is moving some of their symptoms, but not everything. So it's not moving their anhedonia very much. It's not moving their quality of life, which are the ones in the middle, very much. So it's not causing a really rapid change in their overall kind of occupational and social functioning, which maybe is not surprising after only 3 days, but it is changing their depression, anxiety, and stress, which are the last 3 points on the right. And also in kind of a middle range, their positive affect and somatic symptoms and general distress from the MASQ. So there's certain symptoms that are really moving pretty rapidly, and we are seeing changes over just the 3 days focused on the subgenual target in depressed patients. Focusing on the amygdala target, we also just kind of on the first steps of testing out whether we can influence fear response in a fear conditioning context. And so this is a response to a CS plus that predicts an electric shock. And at acquisition, you can see that the skin conductance level is pretty high. Following extinction, between the acquisition and the extinction, this is within subject's design, we did a single round of theta burst either to the inferior frontal gyrus, the positive connectivity with the amygdala, or to the subgenual target. And our expectation was that the amygdala target was going to help augment fear extinction more, which it turns out to be the case. So we're excited that the pathway is something that you can actually modulate, and you can actually move a behavior that's relevant to anxiety through that pathway. Another kind of point I think that's worth making is that if you were to kind of convince our imaging colleagues that something could be added with TMS, I think working at mapping different stimulation sites and what actually happens in the brain, and then also relating that to functional outcomes that you care about is a critical step. So I think that there's still a lot of complexity around neuromodulation approaches, and some of the other speakers spoke to this about what kind of thresholds do you use, what stimulation level do you use, even what the dose is of delivering repetitive TMS to affect a change in a patient. So those issues notwithstanding, I think that those can still be built off some of these basic mapping studies to say, hey, I think I found an individual pathway for this patient that really lights up a specific network, and then going forward with modulating that and having some flexibility in how you modulate that, I think would be the next kind of step up in really showing that novel treatment is worth pursuing. So in conclusion, I think that we're making headway into making neuroimaging more clinically actionable. there's a lot of controversy around doing imaging in a similar way, different groups having different results using the same imaging data, the reliability of something like resting connectivity or a task fMRI. So all these issues, I think, as we're making progress in those domains, they directly apply, I think, to developing better targets for doing TMS treatment. And I think keeping a close eye on the imaging community and the best sort of actionable data we have for making reliable imaging-based targets is going to improve clinical outcomes. So I feel pretty confident about that. And I think that an initial step towards that direction is saying, hey, I think we've mapped the pathway pretty well, and we're showing that it can move behavior, it can move symptoms, it can move brain areas that are all relevant in neuropsychiatric conditions. And that's still based on like the best imaging literature that we have and translating that into doing TMS in better ways. And so with that, thank you. It's a very good time to join a lab and learn TMS or other brain stimulation. It's a good time to apply for grants and figure out the methods that work the best. I think combining things like artificial intelligence or deep learning with making better models of E-fields and evoked responses from TMS and doing things like online, very quick, rapid, individual network mapping and applying TMS in the scanner while doing that. I think these are all fun things that my lab and with my collaborators are pursuing that I think would be really informative about how the brain works and how to make better treatments. So I have a great group of people that all have really excellent skills. There's also always a postdoc slot open with me. We have a lot of data collected by now, and we need more bodies to join in and help us towards exciting new research with TMS. So thanks for your time.
Thank you for your talk. And we have a few questions. First, in your mini dose assessment, are there any thoughts of adopting more stimulation sessions? For example, 10 stimulations a day for 5 days or something like that?
Yeah, yeah. So the initial study was meant to show that there's some movement in symptoms and movement in the brain response to TMS over scale that's clinically relevant, but not to go the full kind of gamut of doing a new treatment. At the same time, in parallel, we have a clinical trial for PTSD where we would give them 2 weeks of the same kind of double dose treatment. And we see that the symptoms move pretty well for the patients there, and that there's a resting fMRI predictor and a cognitive battery predictor. So we're learning something about a bigger dose that's more close to an actual clinical trial. But I think there's also some fun things you could do if you don't expect that you know the change is going to last very long, to mess with the circuits and test behaviors and repeat the experiments with different conditions. So that's another, I think, important avenue.
Okay, the next question. Would it be interesting to use a stimulation target based on the resting state connectivity of both deep regions of interest?
I think that's true. And so actually one of the frustrating things I'm dealing with right now is trying to find a good control site, because by chance, you know, if you choose one not based on resting connectivity, if you just say, okay, I'm going to hit motor cortex or vertex or something like that, and then post hoc you look and you say, oh, where I happen to stimulate already had high connectivity with both targets, then it's a little bit tricky to disambiguate the contribution of resting state and whether it guides you in from the baseline data in where you stimulate. But absolutely, these, like the subgenual and the amygdala, absolutely have overlapping targets, and I think it would be totally reasonable clinically to hit places that have overlapping kind of connectivity with networks that you believe are representative of specific symptom clusters.
Thank you. And the final question, how sensitive is TMS coil positioning with regard to different functional network targeted?
I assume that it's sensitive. However, it's really challenging to fit all of our equipment in the MRI head coil. And so rather than trying to optimize the coil position, we measure the coil position that we're able to achieve, and we're calculating E-fields based on those to see how important it ends up being, especially in the distributed evoked response that we really care about. So what we've seen so far is similar to what Joel brought up in his talk, is that the distributed response to TMS doesn't seem to be related to the local E field in our hands with the limited kind of scope of what we've collected so far.